you have no conflict of interest. Okay. So he will enlighten us about his recent work in the institute, in the lab. So Ilio, the floor is yours. You have about 20 minutes. And then you will have, I'm sure, plenty of questions. Thank you very much again for being with us. And the floor is yours.
Many thanks, Mohamed, Rocco. So today we'll talk about some key features of cancer stem cells. I will focus in particular on immune evasion, as Sarah has already talked a little bit about chemoresistance. And I will show you also some application of the 3D cellular models in these fields. So just a brief introduction. As you know, the tumor system is a heterogeneous, as a heterogeneous population of cancer cells, in some cases organized in a hierarchical fashion with the cancer stem cells at the apex. These cells shares with the normal stem cells the capability to proliferate indefinitely and self-renew while generating a differentiated progeny. So cancer stem cells have been linked to tumor insertion, progression, and metastasis, and in particular to the therapeutic resistance. So it's their targeting is really mandatory for efficient anti-cancer therapies. So the model has been extensively revised, including incorporating some modern concepts in cancer, such as the evolution of cancer stem cells, which can also acquire mutation or karyotypic aberration over time and space, and also the plasticity of the immune system with the interconversions between among states, and also the possibility of fully differentiated cells to differentiate. These are several main implications, to which are that the cancer stem pool within a tumor is more heterogeneous, as previously thought, and that the progeny can also have some degrees of tumorigenicity, at least theoretically. So now the model implicates the presence of some very few defining hallmarks, mostly tumorigenicity and stemness potential, with other variable features depending on tumors type, but also specific tumors, on the origin, frequency, marker, hierarchic rigidity. So there are also some emerging markers, hallmarks, which we are studying in our lab. I will focus, as I told you before, on the immune escape of cancer stem cells. I will show you some results in the mouse models, and then I will switch on 3D culture, in particular on our collection of steroids. So these cells, as we and others demonstrate, these cells has an immune-evasive nature. In this slide, I'm presenting an experiment in which we used microfluidic devices, in this case, 2 chambers. So the red one is the set of red one is loaded by immune cells, splenocytes, pre-stained with specific fluorescent markers, viable fluorescent markers, and the external chamber, the green one, is loaded with tumor cells, cancer cells. So this with this device we can monitor the interaction between a splenocyte and a cancer cell. In particular, we can track the movement of splenocytes which are stained. In this specific experiment, we isolated non-cancer stem cells and cancer stem cells fraction from urine models, and as you can see, cancer stem cells are really low attractive, attract very lowly, very inefficiently immune cells. At longer time, they even move, meaning that they have a higher migration as compared to non-cancer stem cells. This low immunogenicity has been confirmed in vivo, in xenograft, again of the different fraction of non-cancer stem cells and cancer stem cells in immunocompetent and immunodeficient mice. If you we see the graph, so we can really appreciate that cancer stem cell fraction is more immunogenic. The percentage of tumor is less immunogenic. The percentage of tumor-free mice is really lower than the non-cancer stem cells. And this is only in the immunocompetent mouse. We observe no difference in immunodeficient mice, which means that these cells are really very low immunogenicity. We also demonstrated that they are resistant to immune cell death inducers and immunotherapeutic and other immunotherapies. And I don't want to enter into many details, but we observed also the mechanism of immune escape, which is based on the decreased T cell and the dendritic cells and T cells activation, infiltration, as well as T cell exhaustion or the infiltration by some immunosuppressive cells. This has been demonstrated by our lab and also other labs. So as I told you, I will switch on the human system, which is more interesting in the context of the Congress. Sarah already explained this slide, and yesterday I want to explain technically the slides. Anyway, we have a very large collection of steroids and also organoids. Steroids are a little bit more simple to handle for immunogenic related experiments. So in these slides, I represent a table. Each line is a single cell lines, each one derived polyclonally from a single patient. And as you can see, we have characterized this panel at the genetic level. I just want to focus some characteristic. So the panel is divided according on the basis of the diploidy. So in pale blue, the cells that have a diploidy close to diploidy. Hereafter, I will dub them diploid for the sake of simplicity, but they are not perfectly diploid as a whole cancer cell, of course. We have also non-diploid cells in pale rose. When I refer to non-diploid cells, I'm referring to cells that have a number of chromosomes higher than 50. So as you can see, we have an important and considerable percentage of cells that have this chromosome overload. They have supernumerary chromosome. This was expected. This is, of course, in accordance with what reported in literature. And these cells are, as you can see, also several, especially a higher chromosomal instability level. Considering diploid cells, we show that a very considerable percentage of diploid cells, so pseudodiploid cells, display microsatellite instability, and this also was expected. So our panel is very, we are confident of our panel, and as I told you, we use this panel to analyze the immunogenicity and the immunogenic immune system related parameters. I'm showing you some these parameters, which is the expression of HLA-E. And I think that you know this is part of the antigen presentation machinery. So cells and cancer cells are often deficient in this presentation machinery, which makes them resistant to immunotherapy and to immune cell response. As you can see, we observe a heterogeneous expression level of HLA-E with the non-diploid cells, so cell spheroids with a chromosomal overload having a higher level of HLA-E, and very interesting, the diploid cells with the MSI were almost all deficient they have absent or very low level of HLA. It's very interesting considering that this subset is a considered for an immunotherapeutic strategy with the immune checkpoint blockers. So we uncover this unique mechanism of cancer stem cells in our patient-derived models, which is related to the fact that these cells have a low immunovisibility to the immune system. They hide to the immune system. We have recently reviewed these features. We are in this violet part, the so-called camouflage. The cells hide from the immune system, and APP defects, so antigen presentation defect, are among the most relevant camouflage mechanisms. So to conclude this part, we observed that chromosome overload, so an increased number of chromosome amplified seen as expected, but also enhanced the immunogenicity. And we also confirmed the data we already presented, for cancer stem cells. Laura is in the audience, and we worked a lot on this part of the research, and we were happy also to recapitulate these results. So our idea was to basically boost SIN and analyze if we could revert the immune escape of cancer stem cells. We are using several mechanisms to boost SIN. I'm just show you these mechanisms, which is quite advanced in our study, which is based on whole genome duplication. So we force cells to duplicate entirely these genomes, and this leads to increase the SIN, as we showed, and we confirmed also in these models. So the strategy was to establish ploidy clones with different ploidy, analyze the immunogenic potential in vitro and vivo, and then try to characterize and identify specific SIN-associated target that can be used for increasing immunogenicity of cancer stem cells and for clinical development. So very briefly, we used two strategies. The first strategy is a very classic one. We picked diploid cells and we treated them with antimitotics, which forced them to abort mitosis, and then subclone these clones, subclone these cells in diploid and hyperploid clones. In this case, diploid clones, of course, they are pseudo-diploid clones, and hyperploid clones are clones that have a double DNA content as compared to the parental ones. I dub them hyperploid for simplicity. Keep in mind that it means that they have a double DNA content, that they are almost near to triploidy. We have some, I don't want to explain more, but we had several difficulties in this, the optimization of this strategy. We succeeded just 2 out of many multiple essays, months and months of effort, but this also helped us prompt us to hypothesize that the cancer stem cells have a unique control ploidy that limit ploidy change that we are studying that. The second strategy is very simple. We have some cells which which spontaneously undergo whole genome duplication, and we just isolate this fraction. So very briefly, we have several pairs of diploid and haploid clones from only 4 primary cancer stem cells, and these are the cells we used to analyze the immunogenic potential and the impact of SIN and whole genome duplication in the immunogenic potential of cancer stem cells. We take advantages of humanized mice model, in which we incorporate the human immune system by injecting histocompatible PBMC, histocompatible to the human cell, human spheroids we are injecting. As you can see, in violet, hyperploid clones grew less efficiently than diploid clones in humanized models. I don't show you the situation in immune deficient mice, but in immune deficient mice, we observe no differences. When we analyze the ex vivo tumors, generated tumors, as expected, we observed that the hyperploid clone had a higher immune infiltrate, encompassing activated immune cells or exhausted immune cells. which means again that also in our model, the increase in ploidy leads to an increased immunogenicity and recognition, immunovisibility, and sensitivity to cytotoxic T cell to the immune response. We confirm this data also in vitro. This is a macrofluidic device called that competition. In this case, the immune cells are pristine and loaded in the central chamber. The external chamber are loaded with diploid and hyperploid clones. And we follow the movement of immune cells. As you can see, most time, but not always, they go towards hyperploid clones, which means these clones are more immunogenic. We have some difficulties in the reproducibility, which means that we need really in vivo condition to fully appreciate the enhancement of cancer stem cells immunogenicity. Last part, we analyzed the mutational and antigenic landscape, but also the proteomic and transcriptomic landscape. I will not show you the result on the first part because we didn't find any significant results in mutation on neoantigens, which means that tetraploidization is not a mutator phenotypes. But we observed very significant and interesting differences in the zinc genome. The transcriptome and the proteome. Here I show you just the proteome with the reverse phase protein arrays with more than 200 antibodies. We observed that hyperploid clones display the regulation of several protein compared to the near diploid clone, as shown in this diagonal plot, especially for what concern mitosis as expected, cell metabolism, and apoptosis. We focused in particular on apoptosis because we could validate these results. And we have two important results with which I concluded the presentation. The first one is that the hyperploid clones were more sensitive to gram enzymes as compared to diploid clones. It was shown here in PI staining. So we observe increased apoptosis in hyperploid clones. So this is important because gram enzyme is released by effector immune cells together with the perforin, and is responsible for the killing of tumor cells once they are recognized by the immune system, which means that cancer stem cells in general have this mechanisms of cytoprotection to cytotoxic T cells, and the duplication of the genomes increase sensitize them to gram enzyme. We are studying the reason for this result, and we are in the blue part of our immune escape mechanism. It's a cytoprotection mechanisms with the defect in the immune synapse, and so in the defect in cytotoxic T cell killing. The second result was related to the fact that we observed that diploid cells are more prone to undergo apoptosis, mitochondrial apoptosis, than tetraploid clones, hyperploid clones. So we have several studies. I don't show you because it's very long. It's a very long effort, but in any way, what we observed that this makes them more sensitive to anti-apoptotic BCL2 inhibitors. You know that BCL2 is involved in mitochondrial apoptosis. We have anti-apoptotic and proptotic BCL2 protein. We observed that the diploid clones are more sensitive to MCL1 inhibitors, but also to navitoclax and venetoclax, which some of them are also used therapeutically. More importantly, we observed that in a humanized model that the inhibition of MSL1 increased cell death exclusively in the presence of the immune system. So if you compare in this experiment, we xenografted humanized or immunodeficient mice, and then we treated them with this inhibitor. And as you can see, diploid cells treated with the MCL inhibitor decrease their growth only in humanized condition. I don't show you results, but in tetraploid condition, we observe no differences. So to conclude, we observe that deployed cancer stem cells are microsatellite unstable, and these cells have defect in antigen presentation and processing machinery. Colorectal cancer stem cells have a strict body control, and the duplication of the entire genomes is a strategy to boost the immunogenicity and to increase the sensitivity to cytotoxic cell. And lastly, anti-apoptotic BCL2 inhibitors increase the immunogenicity of deployed cells, and this can be used for anti-cancer therapy. So I want to thank my group, Gwenola, of course, Gwenola, but also Luca, Sara, and Claudia, who performed most of the experiment I showed you, and of course my collaborator Antonella Sistico. Many thanks.
Thank you very much, Ilio, for that was amazing. Thank you for this amazing presentation and talk. So first of all, I would like to let you know that as a human geneticist, so what you have presented is a little bit confusing for me. So it's because, you know, I mean, what is fatal for a cancer cell, so like the human, the genetic instability would be also fatal for the normal cell. So I'm assuming that, of course, what you are trying to mention here is, or to prove here is that this is something that could be, or maybe you plan on using for specific therapies. So how are you going to deliver, or what kind of drugs, how the drug will look like if you move forward with this, actually with this idea, and how can you be sure that it's not gonna affect the rest of the normal cells? I hope it makes sense.
I didn't show you the profiling with it, in which we demonstrated that cells are more prone to apoptosis, which means that you can use some drugs really at very low concentration.
Mm-hmm.
This is really the reason why we believe that this can be very interesting. So we can use these inhibitors, which are really very effective, and of course they will also affect normal cells, but we are trying to use them at very low concentration, which means that these cells, that this can be an important strategy. Another thing, and especially we are focusing on cancer stem cells, and this is a problem with these inhibitors, is that they are low, they have low effectivity. So we are more bothered about the fact that this inhibitor alone doesn't function too much, and then effect on normal cells. But in our hand, at least, they have this anti-cancer stem cells effect at very low concentration, in which we believe. Well, of course, the problem with normal cells, they have a very big problem, and you are right in your remark, which is a remark which concerns all chemotherapy or immunotherapy.
Perfect. Thank you very much, Ilio. So now I open the floor for the rest of the audience. So your dear friend, Dr. Jma, would like to ask you a question.
Ilio, I have actually two questions, basically two questions. So your model of predilection is colon cancer, stem cell, but colon cancer. Do you think we'll find the same thing using other model, for example, breast cancer cell, or sarcoma, or whatever? This is the first question. And second question, you understood that all this was also dedicated to the audience here. So the second question is, you show us spheroid, and in my hand, tetraploid and hyperploid cells have difficulty to form spheroid. I mean, have difficulty. I could form spheroid, but easier easier using diploid cells. Do you face the same problem in the lab or not?
Okay, first of all, other models. I mean, we are focusing on cancer, on colorectal cancer, basically because we have a large collection of cells, and this is the reason why we are focusing on this cancer. The other reason is that concerning spheroids, they come from patients which were not treated, and of course we are also trying to explore this this result also on treated cancers, especially on the metastatic background. So basically, we didn't try with other type of cells, which would be a little bit tricky and difficult because we have to produce tetraploid, which is a really very complicated task, as I told you. But I mean, our project is to. You talk about sarcoma. We have some collaborators and also prostate, and we want to try this. Concerning spheroid formation, these are already spheroids, which maybe make it simpler to form spheroids. So I don't know. Maybe you can talk with Sarah. We did some things with the mouse system and the spheroid chips. I don't know if they have more or less difficulty in informing them, and so I cannot answer your question in other models.
Okay, so Sarah is here. I can deal with her.
Yeah,
Question from the audience. Question, yes. You have question, please come to the. So Samantha, she is the global biology technology manager representing Merck, and she has question for you. So you can.
Yeah, so I have a question about your fluidic chips. So we're developing in-house, and we're having a lot of problems with shear stress. So do you guys see a stress response in response to media changing in your fluidic devices? And if not, how are you getting around that shear stress?
I mean, you touched a very important point. I mean, we have several problems in this culture experiment, especially in vitro. Not only with shear. I mean, these cells grow in different conditions. If you think that the cell needs serum and cancer stem cells need to be cultivated without serum, and so we have some problem with the reproducibility. Not technically. I mean, we have not this kind of problem you told us, but most scientific problem. So we have some difficulty in the reproducibility, which is not the case in vivo. This is the strange result, but I mean, we believe in this result because we have in vivo evidence, because only if in vitro evidence we could have some difficulties in trusting results. But I mean, not sure problem. I mean, and we worked with some expert in microfluidic devices, so they are not commercial ones.
Thank you.
Any other question from the audience? Yes. Last question, and then we.
Can you please cite the main techniques used here? Because I didn't understand a lot about the techniques used. And also, at what stage of cancer occurs this aneuploidy? Is it possible to use in diagnostic of cancer?
So I'm sorry, I didn't catch the techniques. We were referring to the microfluidic device or other techniques.
The techniques you described, the distance and when you did some tests and measured the distances.
The microfluidic device technique. So basically, you have some capillaries. You load cells in the chamber, and you follow by video microscopy in the capillary how these cells move towards cancer cells. Is this the question?
Yes.
Sorry.
What was this technique? Sorry.
It's a video microscopy, and you have this microfluidic device. So basically, you follow cells. The immune cells are pre-stained with fluorescent dyes, and you just follow and track the movement and count the interaction times. So this is now it's very common techniques. You can find also commercial microfluidic devices and some methods also online for also with that. From our collaborators, which is Luca Bosinaro. I can give you some more information. Concerning the second question, I honestly didn't catch the last part of your question. Can you repeat it?
Okay. At which stage of cancer this aneuploidy occurs in cancer stem
Okay,
cells.
I mean, this is a very, very complicated question. I mean, all
We
stages.
can use in diagnostic.
It is at all stages. I mean, there is a paper in which they analyzed in which moment you had the spontaneous tetrabolization in cancer. And I mean, you can find it at the early stage or at a very late stage, depending on on cancer type, but also depending on patients. What is clear is that a considerable number of cancer undergo whole genome duplication. Considerable number of cancer at seen, but I mean the exact moment is very complicated. Our hypothesis that can be a pro-oncogenic event, but in some cases very late in tumor formation, which makes it a little bit difficult to be also as a diagnostic marker, of course.
Yeah.